What am I doing? So, my role in the lab is very much as a basic scientist. I am not trained as a medical doctor. I'm trained as a neuroscientist. I make mini brains. So we can take cells out of animals or out of humans. We can put them into an incubator under certain conditions, and they will form spheres that I like to call mini brains. This image at the top here is an electron microscope image, scanning image of the outside of one of these spheres. You can see these different cells on the surface here. And this is how it arranges. But let me just tell you how we make this first. So basically, we take cells from the source, we dissociate them, and by that, what I mean is we take a piece of tissue, we break it up so that all of the cells are individual, they're no longer stuck to each other. When we do this, we lose quite a lot of cells, but the cells that we have left are probably stem cell type cells because they're more robust to this kind of manipulation. What we do with those cells is we put them as a single cell suspension, as we call it, so all separated out, into this liquid food, into an incubator. It's not any old incubator though. An incubator will keep something at 37 degrees C or keep it sterile, but my incubator has a huge platform in it that rotates like this. Very slowly, well, about that speed, but nonetheless rotates. What happens when we put our cells into there is that they're all forced into the center of the dish in which they're kept, and they come into contact with one another. And when this happens, they start to form small spheres over the first 24 to 48 hours in the incubator, and then after a couple of weeks, they're probably a fifth to a quarter of a millimeter in size. And this is what is represented here. Over that period of time, all of the cell types in the brain can be found in the sphere, and so it does represent a brain in many ways. It has synapses, which is something that the cells use to communicate in the brain. That's a very important part. It has myelin. These are arranged like we see in the brain with nodes of Ranvier. So it's a recreation of the brain. There are many things about this that are obviously very different to a person or an animal, and that actually means that we can manipulate it more and do more exciting experiments with it. So what we can do is we can look at the spheres in many different ways. What we've done here is we've actually stained fluorescent red the nerve cells in the sphere. So you can see there's a very complex network of nerve cells in here. And this video just shows you that these are slices taken going down through the sphere. So you can see that at some areas of the sphere there are very dense, like here, and then other areas there's actually a hole in the middle. This is characteristic. There are other cell types in that hole to show you that it is actually arranged spatially. Again, we can do the same thing. Label, this is actually for a dying nerve cell or a damaged nerve cell, and this green is for myelin, and this is a sphere which has been treated to, under conditions fairly similar to multiple sclerosis, but obviously in the incubator. This image here shows dying cells. So what we can do with this kind of image is we can count all the cells in blue, which is all the cells in the sphere, all the cells in red, which is only a portion of them, and we can know how many are dying under different conditions. So we can manipulate this in many different ways. Just to say thank you very much for your attention, and I hope you all have a great day.